terrific. So let's move on now to the third speaker, Dr. John Marshall, who's an assistant professor in biostatistics and epidemiology at UC Berkeley. And he's going to talk to us about genetically modified mosquito strategies and disease modeling to control malaria in sub-Saharan Africa. So Dr. Marshall, the floor is yours.
Okay, thanks very much for inviting me to speak on this webinar. And it's been really interesting to hear some of the presentations from these earlier two projects on the Wolbachia and the Riddle, genetically sterile males. I'm going to be speaking about gene drive systems and their potential use to control mosquito-borne diseases. And some of the involvement I've had in this project since beginning my PhD around about 10 years ago, firstly on the public attitudes to genetically modified mosquitoes with gene drive systems among people in sub-Saharan Africa. I'll talk briefly about the relevance of the Cartagena Protocol to these systems, and then my specialization is developing mathematical models to understand the spread of transgenes and the transmission of vector-borne diseases. So I'll talk a little bit about the relevance of that to trial design of genetically modified mosquitoes.
Dr. Marshall, do you have slides to share with us?
Oh, yeah. Is that not sharing right now?
No.
No.
Do I need to click on something to get that sharing?
Yeah, you go at the bottom of your screen. Are you on the Cisco WebEx Meeting Center?
Yeah.
You see the quick start
tab.
Oh,
share screen.
Yep.
Okay, that's working now.
There we are. Good.
Okay, thanks. So these are the three things that I will speak about. And first, I'd just like to speak about the public attitudes survey that I did a little while ago. So when I started working on this, people were very interested in transposable elements as gene drive systems, and it's interesting to see the progress that has been made since that time 10 years ago. Now, homing endonuclease genes are of large interest in the CRISPR-Cas9 system in terms of gene drive. And of course, the genetically sterile mosquitoes have been released since then, as well as the Wolbachia mosquitoes. So I was working on these and did a public attitude survey in Mali in sub-Saharan Africa on what people may think about the use of genetically modified mosquitoes that would have a disease refractory gene that causes refractoriness to malaria, and also a gene drive system that would help spread that into the population. And this was the first public attitude survey to this technology in Africa, and possibly could inform future public attitude surveys on similar technologies. We asked residents their perspectives on mosquitoes, on their understanding of heredity, the understanding of malaria, of genetic modification, and then we used genetically modified crops as a lead-in to genetically modified mosquitoes, as we thought people may have some familiarity with this. We spoke to people in rural villages, in urban suburbs, and we also spoke to health professionals, both Western health professionals and traditional medicine practitioners. And we conducted semi-structured interviews. We had follow-up questions from the interviewer and interviewee, and these were translated into English. The interview was conducted in the local language. We spoke to people of multiple ethnicities. The sites were chosen for the variety of ethnicities that they covered. And In each village, we explained to the chief the purpose of the survey and the desire for a diverse sample, so that we didn't only want to speak to the chief and the elders in the village, but we would include them in the survey. And one of the interesting things about this study was that there was a lot of familiarity with selective breeding among the interviewees. They were subsistence agriculturalists for the most part, and they practiced a lot of selective breeding with the crops that they grow and with the animals that they breed. So there's a quote here: if you have a strong male goat, then you may take it to your farm to mate with the female goats. They have strong babies. So that was there was some ability to talk about heredity from that perspective. Also, they had a concept of heredity as it applied to people, where it was a mixing of God and blood, where blood was essentially the genetic material and God was the chance aspect, which enabled us to talk about heredity and genes to some extent. So we describe genetic alteration as a faster way to develop more desirable animals, vegetables, or fruits, but that this method could lead to unknown consequences for the environment. Interestingly, also, there was some familiarity with genetic engineering among the population. So people were familiar with the GM cotton controversy in Burkina Faso, where a few of them commented on the politicians in Burkina Faso making decisions about growing GM cotton without consulting the population. There was the perspective that there were GM chickens that were bigger than the traditional African chickens, although they weren't actually genetically modified. They're just they were just varieties from other places. When we gave the example of genetically modified corn, we said that we could provide a genetically modified corn that would be resistant to insects. That it would, insects that tried to feed on the corn would die, and therefore the crop yields would increase. I think that this is one of the most interesting results of the survey, which is essentially the concept of a trial. So we said we have this genetically modified corn. Under what conditions would you grow this corn in your village? And the corn could have an increased yield. We're not aware of any side effects to health or the environment, but there could be some unknown consequences. And they basically said, well, we would like to have our variety of corn growing here, and then your variety of corn over here. Both of the fields should be the same surface area, and they should be a certain distance away, and we should grow both crops the same amount of time and over the same area, and then compare which produces a better yield. And then we would check to see if there was an environmental side effect and if there was a side effect on health if we were to eat this corn. And if the genetically modified corn produced a bigger yield without significant side effects, then we would be interested in growing that corn, because famine is a concern, and therefore we are interested in something that could increase yield. So there wasn't the taboo against genetically modified organisms that we see in some European countries, and there was an interest in solving a problem that could save lives in the event of a famine. And also, it's quite a scientific process that they were interested in. So we led into a question regarding releases of genetically modified mosquitoes. We said, imagine an organization from a foreign country claims they can provide you with a mosquito that has been produced by this method of genetic modification that is able to reduce the burden of malaria in your community. A representative from the UN tells you that the mosquito has no known negative consequences for the community or the environment, They do tell you, however, that there could be unknown negative consequences. Under what conditions, if any, would you consider it acceptable for the foreign organization to release these mosquitoes in your community? And one of the interesting things in response to this question was that a large number of people again referred to this concept of a trial. So many of them said, I would have to see an example of GM mosquitoes reducing malaria in another village before I believe this claim. In agriculture, you have to see something before you believe it. So quite a scientific criteria that they would want to see evidence that this has had the intended effect in another place. And there were the occasional respondents who said, we would want to be an example for other villages, but most people said they wanted to see it somewhere else, so, usually in a similar environment. And there were a number of other things that people referred to, laboratory experiments, access to experimental results, mosquito strains captured from local environment. They would want bed nets provided to protect themselves against the mosquitoes, an education campaign, approval by majority of the community, and another concern was that the community would have to pay for these mosquitoes, so they were concerned about the cost. So taking the results of this qualitative survey, we then went on to propose how you could go about conducting a quantitative assessment of attitudes to releases of genetically modified mosquitoes. Those are published in an article here. And one of the benefits of the descriptive survey, which I just described, is that you can get an idea of the range of responses that people have. So generally you want to precede a quantitative survey with a qualitative one, so you have spontaneity, freedom, and a deep understanding of what people think. But it's very costly, and it doesn't give you an idea of the quantitative nature of the responses, i.e. the number of people who think a certain thing. So the quantitative surveys are faster. You're already committed to your response options, but provided that you've enumerated those comprehensively in the qualitative survey, then they can be very effective. So you want to identify the general themes, meaningful questions, and that range of responses. And a number of groups are involved in conducting some surveys along these lines in countries where these projects are moving ahead. So studies like this can help to inform those surveys. So since then, people are no longer thinking about transposable elements, but in terms of gene drive, one of the most promising at the moment is the X-shredder homing endonuclease genes, where a homing endonuclease gene is on the Y chromosome of mosquitoes, male mosquitoes, and it shreds the X chromosome in male gametes. And as it shreds the X chromosome, the majority of the gametes in male mosquitoes are Y, which means that as the male contributes Y, then most of the offspring will be XY, which are males, and the gender ratio is biased towards males. And this not only leads to population replacement or gene drive, but also to population suppression, because as the Higgs spreads into the population, it causes an abundance of males and therefore no females to produce offspring. So the X-shredder can spread into a population, but then it can also spread into neighboring populations and cause the cascade of population crashes. And in this case, the Cartagena Protocol on Biosafety has to be considered because it applies to the international movement of living modified organisms with possible adverse effects on biodiversity or human health. In this case, the effects on human health would hopefully be positive, but you would have to consider eliminating a species potentially as a possible adverse effect on biodiversity. And genetically modified mosquitoes do fall under the definition of living modified organisms. So in this case, the Cartagena Protocol states that it applies to the international movement of these GM mosquitoes, and these mosquitoes are capable of crossing international borders on their own because they're capable of spreading from one population to another without respecting international borders. There was a concern about Zambia's ban on GM food aid in 2002, despite conditions of famine. Although it's hoped that now that Zambia and a lot of other countries have a functioning biosafety protocol which relates to genetically modified organisms, that will be less of a problem. And also due to the fact that genetically modified mosquitoes are for health, that applications of genetic modification for health will be more widely accepted. But it's broadly understood that these technologies will require a regional agreement rather than just a national agreement if they involve the ability to spread across borders. So I'll talk now about the mathematical modeling and its ability to support field trial design. And we've heard about Riddle and Wolbachia. So releases of genetically modified mosquitoes, it's most appropriate that genetically sterile males are released first, as they are at the moment. Then you would have systems that can be confined to isolated populations. So the biology can be, with the efficacy and the safety can be determined in a confined way. And then you would have invasive systems like the X-shredders, which have the potential to control disease on a wide scale, but also have stronger regulatory concerns. So one of the questions that mathematical modeling can help to inform is the safety and efficacy, and related to that is the ability to confine a release of genetically modified mosquitoes to one population so that you can observe its effect in that population without it spreading into neighboring populations. So here we have 2 villages in Mali separated by about 7 kilometers. Ecological experiments suggest a migration rate between these villages of about 1% per generation. And the goal would be to have the mosquitoes spread into this population without spreading into the neighboring population. And this is data from drive experiments on an underdominant system which displays threshold properties. So in a lab that I was working in at Caltech, Bruce Hayes' lab. And if you release the system at frequencies above around 25%, it spreads into the population. But if you release it at frequencies below that, then it's eliminated from the population. So the hope is that if you have a system like this, then it is confinable to isolated populations. It will never exceed the threshold frequency in one population and in the neighboring populations, but it will exceed the threshold in the released populations. So this is the a mathematical model fitted to the drive experiments in the laboratory. And if you apply those to our 2 villages in Mali, in Banambani and Donagibugu, which were in the last slide, with a 1% migration rate between those villages, then you can see that if you release this construct at above 50% in the release village, it will spread to fixation. And then you can actually dilute it with wild types in the population, and it'll be sub-threshold now and then eliminated from the population. But in the neighboring population, it never exceeds the threshold, so it's just eliminated from the population. or it never spreads into the population. So this has the kind of dynamics that we would like to see in confined field trials, where it spreads into the release site, it can be reversed, and it's confinable. And mathematical models can help us to determine several aspects of these studies. For example, which is the best confinable system that we can use here in terms of efficacy of malaria control and confinability. How many releases and what size of releases are required to see the desired effect? And should we monitor disease prevalence in people, or the disease prevalence in mosquitoes, or the transgenic status of mosquitoes in order to monitor these trials? Those questions can be addressed by mathematical modeling. And also we can assess which field sites would be most well suited to this type of study. So would it be islands off the coast of Africa, or could we look at villages as we had on the previous slide? So mathematical modeling can support trial design before the trial is carried out to determine the optimal intervention, to compare trends in disease prevalence in the control arm and the expected trends in the experimental arm as well, and also the magnitude of the intervention that's required to see this effect, and how long the trial has to be carried out for. Then during the trial, we can monitor the development of the trial in both the experimental site and the control site. And modeling can help to check whether the desired effect is being observed. It can trigger correction, mid-course correction to our interventions during the trial, and to assess whether the likely impact is going to be observed in the end. And this all reduces the risk of an inconclusive trial happening. And then after the trial, we can compare the predictions of the mathematical model to what was actually observed. If they're in agreement, then we have validation of our understanding, and that's encouraging for future wider scale releases. If there's disagreement, then we can work out what the source of the discrepancy was, try to do that, and then improve the model, taking into account the discrepancy to see whether we can mimic that result. If there was a negative result, then we can try and understand what that negative result was due to, and whether we would need a larger or longer term trial to see a positive result. And this is my last slide. If we have done successful trials, then we can take that mathematical modeling framework and apply that to the wider scale releases, which would be implemented for homing endonuclease genes or CRISPR-Cas9 gene-derived systems, where the goal is disease control on a very wide scale. But before we were to do that, we would want to make sure that we're confident with the efficacy of the intervention and safety of the intervention. But if we were looking forward to that beyond confined trials, then we could ask questions such as, which is the best genetic system to carry this out in terms of disease control and safety on a wider scale, and what are the best release strategies for these systems, and questions related to the cost effectiveness of these interventions. So I'd just like to say thank you at this point to my collaborators along the way at UCLA, Caltech, and Imperial College, as well as UC Berkeley, and we had funding from the NIH as well during this study, and the Medical Research Council in the UK.